Today is the first time in my career that I will present sequencing data that isn't from Illumina. And I reached out to Ultima and the Ultima team about a few years ago because I really thought that maybe their technology could help us address some really fundamental questions in genomics, and that's what I'm going to tell you about today. I think one of the fundamental questions in genomics is how we move from correlation to causation. So this is an output of a single-cell RNA-seq experiment. You've all seen heat maps like this. We can look at a per-cell resolution and understand detailed molecular signatures of genes which change between health and disease. We have incredible resolution to be able to do that with functional genomics. What's hard is, what do we do after this? How do we go from this sort of observational correlation-based data towards a more causal and regulatory understanding? How do we know what drives these particular types of changes? All of our labs deal with this. This is a common problem, and what we do is the same as what you guys do, which is we use these databases, Gene Ontology, MCDB. These are effectively dictionaries of signatures where we know what the underlying set of regulatory perturbations were. We know what genes change as a result, and then we try to match those signatures to what we see in our observational data. And the idea is that if we can find a nice match, we can sort of infer causality in functional genomics data. So these are incredibly valuable resources. They're things that we use every day. They're things that your labs use every day, not just for single cell, but for spatial and bulk technologies as well. And these databases are valuable, but they're sort of cobbled together from lots of different technologies, lots of different papers. There's domain expertise. There's not necessarily a unifying framework for being able to create them. And what we've been interested in is to ask, what if we could construct molecular pathway signatures in a systematic and data-driven manner? What if we could find a new way to be able to generate these dictionaries at higher sensitivity and a higher degree of resolution? And that's what one of the, it's a long-term vision. My lab has taken some very early steps towards this work, and we published a preprint just last week or so that was led by Longda, Carol, and Effie in my lab, so you can check this out. Everything that I'll show you is available, and all the data is also available online. And really what we wanted to do was say, can we use scalable single-cell perturbation screens to be able to identify the hallmark targets of our major signaling pathways and also their sub-branches? So there's data and there's experiments, but there's also kind of a broader goal of being able to enable a new scale of single-cell experiments. And we were glad to see that other people in the community saw this. In fact, we got some feedback on our preprint from none other than the Genomics cow. If you know, you know. This is a Twitter account that posts some very hot takes and very insightful takes on genomics. If you know who this is, also please tell me because I'm very curious. And what they point out correctly is that the size of single-cell experiments is increasing even faster than Moore's law. And so now it's not just that we can be thinking about sequencing a lot of single cells, but we can be thinking about sequencing a lot of single cells from hundreds and eventually thousands of samples, and that's a very, very exciting vision. So what are sort of the cost drivers that are kind of stopping us or preventing us to get to that vision? And there are three. So of course, library preparation. We need to prepare libraries for millions of cells, and new technologies that enable, for example, combinatorial indexing, or you heard some work previously from Fluent, are causing those costs to drop very quickly, and that's very exciting. We still do need to be able to sequence all of those libraries, and of course that's why we're here today, to talk about the ability to be able to get billions of reads. They don't need to be very long. We don't need 500 base reads. We just need to be able to map to the transcriptome and sequence the barcodes that we're generating in our single cell data. Even 100 bases is fine. But the other cost is actually, if we want to do population scale sequencing, we need to get thousands of samples, and actually the cost for being able to just prepare these samples and obtain them can often be the most expensive part of this workflow. And so actually it's not necessarily true that tomorrow we're going to start doing single cell sequencing of thousands of individuals, but there are other ways of generating large numbers of samples. And that's where we're so excited about these perturbation screens. So you heard some work on this earlier as well, but we were really inspired by this paper. It's a paper called Genome-wide Perturb-seq from the Weissman lab. And what they did was they had a giant dish, and every cell had a different guide RNA knocking out one of 10,000 different human genes. So you do a huge single-cell RNA-seq experiment, and in one giant analysis, you can hopefully learn the function of all 10,000 of these genes in parallel. Now, they did this experiment with 10x in Illumina, and they invested very significant resources in order to be able to do it. And maybe you think, okay, well, they've done it now. We already know the function of all genes. We're done. But of course, the challenge is that they did this experiment in resting K562 cells, and of course, many of the signaling genes and regulators that we care about will only become active in particular biological contexts and different cell lines, and only after cells have been exposed to different ligands. So if we really want to learn gene function, we need to be doing this scale of experiments in multiple biological contexts, and that starts to get seriously scalable, and that's one of the reasons we reached out to Ultima. So the experiment I'll tell you today, we took 6 cell lines across different human tissues, and we exposed them to 8 different signaling pathways. This is a combinatorial matrix. So we generated 48 different biological samples, and in each one of those, we did Perturb-seq. We didn't do genome-wide Perturb-seq in each one of these cases. We focused on signaling regulators. But this is a large data set, and for us, we generated actually a couple of years ago. This was by far the most scalable project that we had looked at. So we have about 2,500 perturbations in total, more than double or triple that if you count number of guides. We sequenced about 4 million cells. This was done with the parsed mega technology with combinatorial indexing to keep our library costs down. And we did our sequencing with Ultima Genomics. So I'm just going to run through some very brief vignettes about what we were able to do with this data. If you're interested, please check out the preprint in the paper, but I want to tell you a little bit about what we did. So this is one data point from our study, okay? We take the ligand IFN gamma, we stimulate HAP1 cells, and we knock out the gene interferon gamma receptor 2, and we compare that with our non-targeted controls. Of course, all of these are mixed in the same dish, and we get a beautiful gene signature of genes that go up and genes that go down, specifically due to this perturbation of interferon gamma receptor 2. So it's a very strong causal, this is not observational data, this is causal data. We have a very clean, beautiful molecular signature. There's no batch effects in this experiment. Again, this is one data point. Our data set has thousands of these different perturbation signatures across cell lines, across regulators, and across signaling pathways. Again, this is not observational data. This is real causal perturbation, dictionaries of causal perturbation signatures. Just as a brief computational vignette, one of the things we've observed and others in the field have as well is that when you do CRISPR-I perturbations, it doesn't work equally well in every cell. Sometimes you get a very strong knockdown. Sometimes you get a weaker knockdown. There's a sort of continuum of the degree of perturbation. There's actually something we can learn by analyzing the data. We released an algorithm called mixed scale, which infers the degree of perturbation strength in each individual cell. And the reason why that's valuable is if we know how well the knockdown worked in each cell, we can actually use that information going forward when we do differential expression. And so we introduce a weighted differential expression framework where cells that had a stronger knockdown contribute more to the downstream analysis, and cells where the knockdown was weaker contribute less. And it turns out that when we use this differential expression framework, that strongly boosts our statistical power and our ability to discover differentially expressed genes by about 30%. So if you're interested, all of this is available online. The mixed scale framework and our differential expression package is all on GitHub and is compatible with our R software package, Seurat. All right, so what about Ultima? How does Ultima sequencing perform? And actually, just to be sure, we sequenced a subset of libraries with Illumina as well. So on the x-axis is basically a pseudobulk RNA-seq library with Illumina. On the y-axis is the same exact library sequenced with Ultima. Each dot is a gene. This is actually data from a couple of years ago, by the way, too. But it looks pretty good. Like we see a very strong correlation. But there is a very small batch effect if we compare Illumina data and Ultima data. So if we take 2 different experiments sequenced with different technologies, there is a small batch effect there, as there would be with any 2 technologies. It's a very reproducible batch effect, so it's easy for us to build models to correct for it. But more importantly, we're not trying to compare Illumina and Ultima. We're trying to compare Illumina controls and Illumina targets with Ultima controls and Ultima targeted cells. So what if we do that? How do our biological insights correlate? And that looks absolutely beautiful. So what I'm showing you here is what happens is if we compare within an experiment the knockdown of our non-targeting controls to our targeted cells, and actually each dot here is a p-value. So you're basically seeing that when we run the experiment with Illumina and we run the experiment with Ultima, we get exactly the same biological insights. This to me was very exciting reproducibility. The difference, of course, as you've heard about extensively today, is the cost that it took for us to run these 2 independent workflows. So we're fully comfortable switching our Perturb-seq sequencing entirely to Ultima. All right, so what do we do with all this data? We have, 1,500 different perturbation signatures. That's a lot of data to deal with. We thought maybe we could look for regulators that perform correlated functions in a way that would enable us to effectively reconstruct molecular pathways from scratch. And that's what we did. We build off an approach called Dialogue from Aviv Regev's lab, which uses multi-CCA, to identify what we call perturbation programs. These are groups of regulators that do similar things and presumably correspond to arms in the signaling pathway. So let me show you what one of those perturbation programs looks like. This is the core perturbation program for interferon gamma. We identify correlations between interferon gamma receptor 1, 2, JAK1, JAK2, and STAT1. All of these regulators are very upstream core regulators of the interferon gamma pathway. When we knock them out, we see hundreds of genes that change in a conserved way across cell lines. This is really a hallmark gene set. When we see these genes change, we know there's a change in interferon gamma signaling. But what's cool about our data is we can go much further than this. We can also start to identify sub-branches or subcomponents of the individual pathways as well, and that's what I'm showing you here. We can automatically identify a second correlated perturbation program. This is a subset of the first one, but it corresponds to downstream targets of IRF1. So this is a sub-branch of the interferon gamma pathway. It's actually repressed by IRF2. And so now if we see a set of genes change, not only do we know is this driven by interferon gamma signaling, but we can say whether it's driven in an IRF1 dependent or an IRF1 independent way. And we return dozens of these perturbation programs against all the different stimuli that we look at. And again, all of these data are available online for you guys to score against your experiments. So how do our signatures compare to things people use all the time? So we looked at, for example, the MSigDB database. This has been cited more than 12,000 times. We've certainly used it many times. It's one of the most widely used molecular signature databases. Looking at interferon beta, for example, we see very, very high overlap between our gene sets and our gene sets, but we discover hundreds of new hallmark genes that are conserved and reproducibly identified across cell types and regulators. So we get a much bigger signature, and this is true for all the pathways that we look at. We discover a couple hundred additional genes, and I'll show you in a second why that's so important. So now what we can do is we can go back to our original problem. All right, well, this is the heat map I showed you in the beginning. There are hundreds of genes that change. We know that in COVID, there's going to be some sort of inflammation, for example, in our monocytes. And if we do sort of an enrichment program analysis on our data, we see multiple pathways come up. We see interferon beta, we see TNF alpha, we see interferon gamma. One possibility is that all of these different pathways are changing. There's some sort of cytokine storm. But the other reason that we're seeing all these pathways come up, and if you guys do go enrichment analysis, you know this too, is that there are overlapping genes in these different pathway sets. And so it's hard to really distinguish one from the other. But because our gene lists are so large and so sensitive, we can not only identify shared genes across these pathways, but exclusive ones as well. So now for the first time, we can go and ask, is there enrichment specifically for genes that are only detected as being relevant to these different individual pathways? And when we do that, we can see that it's not just a broad cytokine swarm of all cytokines contributing equally, but at least in blood and at least from monocytes, we see a very strong and specific contribution from interferon beta, and in fact, we can suggest specific regulators which are driving that as well. This is not just something we see in monocytes. We see this across multiple cell types, and we see, as has been previously observed, that as COVID severity increases, in fact, the response of individuals actually goes down. I'll just show you one other brief vignette. This is now looking at spatial data. This is actually from Joachim Lundeberg's lab. They were looking at a mouse model of colitis, and they were looking at the healing intestine. And so they generated some very nice data where they have, of course, H&E stains that are annotated by a pathologist, and also data with the Visium platform. And what they found is that there's a very specific region of the intestine where they found that wound healing was taking place, where they identified this inflammation and hyperplasia. If we look in our gene signatures exactly in that region, perfectly overlapping with the pathologist annotation, we see a very strong and striking upregulation of our TGF-beta activation score. TGF-beta is, of course, a crucial pathway that's involved in wound healing and regeneration. But if you use any existing gene set or any existing signature, the data isn't strong enough to be able to capture this signal. What's also kind of fun about this from our perspective is this was in mouse. This was in mouse colon. We didn't even use mouse in any of our initial data. But because our gene signatures are learned across so many different biological contexts, that enables us to apply them to new ones that we didn't profile at all. And in fact, one of my favorite vignettes from the paper is our ability to verify that we can determine IRF1 dependent and independent arms of the interferon gamma pathway, but in bat fibroblast cells. So with that, I'll wrap up and I'll thank my lab and I'll thank Ultima. You know, I'm here to talk about our work, but also as a member of this market that's just looking for lots and lots and lots of short reads. And I think if we can do that, that's going to enable us to ask new biological questions, especially for functional genomics. Thanks very much.